Hello and welcome to Ferment-Home-Station, a National Science Week event in Australia for promoting the science behind fermentation. My name is Deepan.
My name is Lenny.
And today we are joined by renowned microbiologist, entrepreneur, science communicator, innovator, and TED speaker, Dr. Anne Madden. Hi, Anne, and thanks for joining us for this exclusive interview for National Science Week.
It's an honor to be here. Thank you.
So you're an adjunct faculty at the Department of Applied Ecology in North Carolina State University. You're also the founder of Microbe Institute. You're chief scientific officer at Lachancea. So your career has been in science and with microbiology, but where did this all begin? Like, where did your journey in science begin?
So I didn't grow up wanting to be a scientist. In college, I was sort of meandering towards a career in veterinary medicine because I liked animals and I liked taking science courses. But through sort of a series of odd events, I ended up with an internship in Costa Rica where I could study whole different systems in the rainforest. And there I got to help scientists track venomous snakes, and I got to collect poison dart frogs and learn about this fantastic jungle of unknown species. And I was hooked on being an explorer and being surrounded by mystery. And then I came back to my senior year in college in the Northeast in the USA, and there was no jungle, there was no sort of mystery of animals around me. And I took a microbiology course. And I found that indeed I was living in a mysterious jungle full of unknown creatures. It was just at a different scale. And so from then on, I've been near obsessed with this microbial cosmos around us.
So we are calling this talk Exploring the Microbial Cosmos. What do you mean by the microbial cosmos?
So when I refer to the microbial cosmos or the biological cosmos, I'm thinking of most of life. Most of life's species exist just beyond our line of sight. These are incredibly small, microscopic creatures that are on every surface we touch. They're floating through the air. They're on every granule of sand. And they are an entire world unto themselves. And this involves everything from our microscopic animals like face mites or water bears to things like fungi or yeasts, bacteria, those that are on our tongue that we're brushing off on our teeth, as well as those that are in sourdough or make yogurt tart, all the way down to viruses that we hear a lot about in the news these days.
That would be my question. What's the difference? I mean, my mother taught me well to always wash my hands, get all these germs off. Like, how do we define that one is bad, one is good? I mean, especially now you said already that sourdough, something we use now a lot. So do I need to wash my hands after the sourdough? I mean, what's the difference between the good and the bad, basically?
Yeah, it's a great question. So we live in a world with about 1 trillion microscopic species. That is sort of a mind-blowing number of creatures, and each of these species are as different as a banana is from an elephant, or even more so. And just to make that sort of a little bit more tangible, if you were to go outside in your backyard or in a park and pick up just a sugar packet's worth of soil, so just this much, a few grams, you would have inside of it more microbial species than there are animal species in all the world's zoos.
Oh, wow.
If you get one more sugar packet of soil, you have a whole new planet's worth of species. So our microbial world is very diverse. And that brings up the question, what is our relationship with all of these different species? And it's just like our relationship with animals or plants. My relationship with poison ivy is not positive. I break out in blisters. I really don't love that plant. But my relationship with strawberries or plants that make chocolate is a very different relationship. And it's just like microbes. Some species do cause disease. Other species are actually helping us make our lives longer, better, and more enjoyable.
Oh, that's great, yeah.
So just taking a step back, for our audience, how would you define microbes? Do all these fungi, virus, bacteria fall under the same group, or are they all microbes?
Yeah, so I think this is one of these like mysteries of science, that the simple questions don't often have simple answers. And so I'm going to use the description of microbes, which is microscopic species, though there's a little bit of wiggle room in understanding what constitutes that. But when I'm talking about microbes, I am referring to everything from viruses to bacteria, archaea, fungi, even microscopic animals. But my own work relates to bacteria and fungi.
Thank you. Yeah, that's a definition I think that boggles a lot. Like you grow up learning this classification of the animal kingdom, and then you struggle with like, is virus a microorganism? Is virus even living? So that's a very, I think, nuanced question.
So is a virus living? Question for the expert.
The questions just went from the very sort of introductory to the hardest questions that perhaps we address as humans. What is life? Yeah. When I'm talking about microbes with different groups, when I'm looking at these different creatures for the purposes of what I'm doing, they are life forms, though they require viruses, unlike other sort of microbes, require hosts. They require our cells or the cells of earthworms or plants to replicate, but they're still microscopic entities that functionally seem a lot like small life forms. So I think that's where I will go as far now.
So you were talking about the microbial cosmos and the different size of things. How do we get a visual of something that we can't even see?
Yeah, so I think we struggle with this when we think about the microbial cosmos, so many species, but it really is at this different scale. And so to help with that visual, I created a hat that has a 3D printed coronavirus particle. And if this virus was to scale, this black hat would be about the size of a bacterium. And so that's the microorganism that you're familiar with in your gut or making yogurt, things like that. If we wanted to think about what a fungus then looks like, something like a yeast, we're at the size of about a tire. It's somewhere almost 10 times larger than this. And if we want to then understand what one cheek cell of a human would be, wearing this hat, we'd have to take 7 giant steps to cross one skin cell. So this microbial world is incredibly small. And because of their size, they've diversified over time so that there are species that can do sort of unimaginable things. There are species that can live in the coldest places on Earth, those that can live in boiling water, those that can feed on sugars and nectar, and those that can feast on plastic, all because there are so many spaces for so many species at that scale.
So you mentioned a lot of microorganisms in sourdough, in yogurt, on our hands. So we might not be aware of all the microbes that we interact with. We might be aware of the ones in our food, but we might not be aware of the ones that we interact with on a daily basis in our homes. Could you, from your experience, give us a few interesting examples of things that we live with all the time?
Sure. So our homes, particularly now, are places we know very well. They are the farthest thing from exotic. And yet inside of them, there are often thousands of microscopic species. But I'm not even going to talk about those species that exist in our house dust or perhaps our pillows and our skin. I'm more interested in those that have helped us create things that we actually love and enjoy. We just don't often trace back their microbial origin. So we'll start definitely with yogurt and bread. Those are 2 things made possible by microbes. And if you're having sourdough and you're having yogurt, that same tartness that you're getting in your mouth is from those microbes. But if we extend what we look at and we look at our first sip of coffee in the morning, a lot of those flavors actually came from microbes that were breaking down the coffee berries thousands of miles away before the coffee berries were dried and ground. And so we're getting a microbial silhouette. And then if we go, perhaps we're still in our morning, we've had that cup of coffee, now we're brushing our teeth. So we're gonna have a prop here of that we're using a toothpaste to brush away the microbes that we don't want on our teeth. There's a microbe that's actually in charge of making our toothpaste gooey. And so there aren't microbes in our toothpaste, but rather in the manufacturing facility, there was a microbe that created this goo, this xanthan gum.
That's amazing, yeah.
And in nature, this is a microbe that produces this mucousy sugar while it's killing plants. But a group of scientists found that, hey, that same mucousy quality will be great if we use it in food to help us make everything from toothpaste gooey to cosmetics to, you'll find it in salad dressing. It's ubiquitous. And we can kind of continue this. So our linen, if we're wearing linen clothing, is made possible by microbes that actually helped break down the flax, the plants that linen comes from, and was a key step of turning a plant into this beautiful linen that's now making our clothing. If you're on antibiotics, or perhaps you have a parent or loved one that's on a cholesterol-lowering medication, both of those medications came from microbes that you can find in our soil. And so truly there's a microbial narrative thread of many of the technologies that we don't even notice we're using.
That's incredible. I didn't know about the xanthan gum story. So in your examples, you talk about microbes, good microbes that we interact with all the time. Yet, for some reason, as Lenny also mentioned, growing up, microbes are germs that we need to wash off our hands, that we need to stay away from, that gives us flu. So why do microbes have such a bad press? And as educators, can we do something to change this narrative?
Yeah, so I think this is a beautiful question because it's a way of providing information to students, to audiences that can actually reveal how much better the world is than it first appears. I think oftentimes when people hear microbes or germs, immediately they think of the last microbial moment that they had. Perhaps it was strep throat, perhaps it was remembering the pain of an earache or an infection or tooth decay. And that's where we often stop the discussion. Oh, microbes, they cause pain, they hurt my loved ones. I don't want to talk about it anymore. But if we dive a little bit further and as science communicators, as educators, and we share some of these other microbial moments that people are having every day, whether it's the xanthan gum in their ice cream that's making it wonderful and creamy, or if it's the microbial origin of flavors of chocolate or coffee, then people will start to understand that their microbial interactions are not just negative and pain. Those are just the ones that have been talked about the most, and they should, right? We need to protect ourselves, but that's not where the story ends. And so I think by highlighting these other microbial moments that we have, we can start building a better relationship with the microbes around us.
Yeah, thank you. I guess on the other side of that spectrum is something like probiotics. And there's ridiculous claims out there about what a probiotic can do, or like it can cure everything right now, if you believe the internet. So there's I think a lot of care should be taken on education in that spectrum too.
I completely agree. So we know that microorganisms are incredibly powerful. We know from really beautiful science, really robust evidence shows that microbes can do remarkable things. And I think that that makes us want to believe any claim that a company may have around what a microbe's doing. And it's really the burden is on us to make sure that we're asking for science and evidence to back up those claims of efficacy. And as science communicators, it's sort of our responsibility to educate populations so that they're informed and enabled to make decisions and to ask the questions of, you say this probiotic can do X, Y, and Z. Where's the evidence to support this claim, so that you can tease apart what's science and what seems to be snake oil or marketing?
So we could, I guess, start slowly getting into the good microbes and fermentation.
Oh, yeah.
Could you walk us through the process of like discovering what a microorganism does, and like what a function of a microorganism is, and how do you find that it might be useful for us?
Yeah, so the best part of my job is being a microbe wrangler. It's exploring new spaces and then trying to wrangle these species into the lab to understand what they're doing, what they eat, what they require, and then what's special about them. All species are different in many different ways. We know this in terms of an elephant has very different characteristics than an apple. And microbes are the same way. It's just about discerning what does this species do exceptionally well or different, and perhaps what is a creative way that I can use this microbe to help with some problem we have as humans. How can we find microbial solutions? And so that's one way. Finding a new species allows for that opportunity of what could you help us with? And that's sort of a guidance counselor approach, sort of understanding what are the skill sets that you have and where would you be great in a career? And then there's another approach, which is understanding, do you have a sort of an obstacle that you want a microbe to help you with? In the past, we were looking for a new yeast to make beer. And so the question became, where in this world do we think that there's a microbe that has already evolved the traits of interest? And so with that question, we took the approach of looking through our understanding of ecology and evolution and finding that wasps, of all places in the world, are a place that you can find yeasts, these same organisms that are involved in beer brewing, bread baking, donut making. And so we went to wasps to help us find yeasts. And from there, it was about sort of understanding which of these many species that can come from a wasp will be most useful for making beer.
So did you find any beer making wasps?
So we did. This is a wonderful success story. So we were on a sort of expedition to find a new brewing yeast. And our goals were humble because we know that while there are yeasts in nature that can make beer, they usually don't create good tasting beer. Because to make beer for a yeast is tough. They have to eat the right sugars in grain. They have to not only produce ethanol, alcohol from that, but they have to then produce enough alcohol for 4 or 5% beer that we're looking for. And then they have to create good flavors at the same time. And that's often the hardest of those 3 obstacles to overcome. And so went to wasps and bumblebees and other insects that are flying around to sugary sources, because much like a wasp and a bumblebee, yeasts like sugary environments. And they seem to be using these insects as airplanes to get from nectar to rotting fruit to tree sap, to all of these places where you'll find ephemeral sugar in nature. From these wasps grew out dozens and dozens of yeast, and then tried to find which one of these yeasts would have the abilities to make beer. And at this point, again, we're hoping that it can make beer. Even if it creates bad beer, that's okay, because it was really an education outreach effort. It was showing that indeed wild yeasts from nature can make beer. But in the process, we actually found a whole new group of yeasts that hadn't been used previously for brewing. And they could make a beer under the normal timelines for making beer. They could make a beer with plenty of alcohol, actually enough for strong beers. And they could make it with really lovely flavors. So some of these flavors include flavors of honey. So there's a compound in some beers called glycerol, it's a type of sugar. And it creates that mouthfeel that you get when you have a really kind of nice bodied beer. And this yeast was producing it. It was also producing aromas that are what we register as fruity and floral. And most impressive of all, it was producing lactic acid. So that same tartness that you're getting in yogurt, this yeast could produce. And so we found a yeast that could make a sour beer in 2 weeks. And if you're from the brewing world, sour beer often involves a whole bunch of different microorganisms, the yeast that can make alcohol, as well as those that you often find in yogurt that are producing the tart notes. It's riskier for a brewer to undertake because those yogurt microbes can go into all of the beers. All of the beers can become sour beers. And it takes a long time. You get a lot of batch-to-batch variation. And so long story short, with the help of these wasps and beers, we found a new way of making better sour beer. And within 10 months, we'd worked with groups to commercialize this beer, and it's now making award-winning beer, cider, and sake in the US and Japan.
That's an amazing story. I didn't know that. Definitely will look differently at wasps and insects now flying around, and wonder if they can make me some beer.
And drop a wasp in our batch.
Yeah.
And so, yes, the subtitle of this work is What Good is a Wasp?
Yeah. Great. Yeah.
I should highlight, this shouldn't be done at home because there are microbes inside of wasps that are also not safe to work with. And so there are caveats to ever carrying this out. But yes, if you see a wasp in nature, you can now thank it for producing better beer flavors.
Out of curiosity, how did you begin this your research in this space of fermentation? Or you mentioned that you started working with microbiology in graduate school. Did you always work with fermentation or food-related microbes?
I didn't. My first research was on understanding microbes associated with polluted ponds and understanding how they'd survived this pollution. And after college, I went to a biotech company where we were searching for unculturable microbes. So those microbes that are in soils or marine sediments and we can't ever grow in the lab. And from those microbes, we were looking for novel antibiotics. And in the process, we discovered novel antibiotics, and this hope is to better meet the needs of antibiotic resistance and the issues that those pathogens are causing. And so from this start, I got a taste for being a microbe wrangler, for getting these microbes from nature and then discerning which ones are good for the application of interest. And so I moved on to graduate school where I was investigating the microbiome of wasps. And now I think we can say, oh, of course, it was to eventually make better beer. But at the time, it was really an interest in exploring a space that hadn't been explored with a microbial lens. These are the paper wasps that we often have right on our windowsills, or those that you'll find buzzing around cans. And no one had really explored them for the microbial species they might be carrying. And so that was part of my interest, was exploration to see what these species could do. And in the process, we got to discover new species and name them. And it led to this work somewhat inadvertently with beer. And I think that that was my first work with fermented foods or beverages. But from there, I started working with coffee makers, with beer makers, and then in sourdough and bread making. And so those microbes that are involved in one fermentation are often used in many different ferments.
You mentioned sourdough, and we are having Carl DeSmit from the Sourdough Library in one of our events, and I know that you've worked with him previously in one of your big sourdough projects. Could you describe how that, what that project was about, and what did you find?
Sure. At the time, I was working at North Carolina State University. I was working with Rob Dunn, and as a lab, we were interested in understanding the mysteries of sourdough. So unlike many of the microbes that we've talked about, this is a microbial community that almost everyone loves. Everyone has a beautiful relationship with their sourdough, even if it's a somewhat frustrating relationship, like a pet, usually has a name. And so this is a wonderful community to explore with people because it still holds mystery. We've been making bread for at least 12,000 years or so, and yet we still don't know all of the species that help make these flavors that we love. And so we worked with Carl and his group to understand a few of the microbial secrets. Where are these microbes coming from in sourdough, and how do these species translate to different flavors in our breads? And so I'll share some of the findings. We worked with a number of professional bakers, and we swabbed their hands as well as their own starters that we asked them to create using the same protocol and using the same flour. And we found differences among these bakers. Even though they were using the same flour, even though they were using the same protocols, they're really different starters in terms of what species were in them, as well as how acidic they were. And when they baked bread with these starters using the same ingredients and the same equipment and the same bakery, these breads were discernible. They had different flavors that related to those species. And we found that a lot of those species were coming from the flour, but some of them were shared with the actual baker's hands. So there's this beautiful, intimate relationship between baker and sourdough starter and flavor. And we are still teasing out some of the results related to that.
Oh, that's amazing. Didn't know that it makes your own bread very unique in that sense. Yeah.
Currently you're working on any super cool fermentation related projects?
Yeah, so I've partnered with NC State, and we're doing a participatory wild sourdough project where we're asking people to create their own starter, and I'm sure you're very familiar with that. And so that's one sort of project. In terms of the sort of wasp beer research, we're delving into other fermented beverages that these new yeasts can provide new flavors to. And so our partners just released a wasp yeast sake in the Japanese market that has sort of lovely slight tartness to it. And then working with different groups associated with the Microbe Institute to make microbial discovery more accessible. And so we haven't launched all of our projects yet, but the goal is really to someday allow people in their homes to discover and name new species and be a part of this exploration. And we're also working with artists and educators to make sure that these are interdisciplinary projects that engage broader communities.
That's very, I'd be super keen to learn more about the Microbe Institute if you want to talk about it, some of your initiatives or how you plan to like put together scientists and artists. Seem to be a very, it'll be a very interesting journey, I'd say. And the outcome would be also very interactive.
Yeah, so I think that the hope is to create sort of a group of people that are all interested in microbial discovery and can bring different understandings to that. So some of the projects that we have include a past project that I worked on with an artist, Amanda Thingbata Hipakaya, where together with funding from the Alfred P. Sloan Foundation, we created an interactive art and science pop-up exhibit that used augmented reality to animate microbes in our spaces around us. And so there's an app associated with this on the website where you can actually have yeast cells in beer pop up and move, and you can learn about them, learn about some of the microbes in our carpet that can produce some of our incredible antibiotics and medicines. And so that's one way of sort of engaging new audiences with this microbial cosmos. Also working with collaborators over at Bard College on participatory science projects that allow students and the general public to sample microbes around them and be involved with that microbial discovery. So there's a whole bunch of different initiatives that we have, and if anyone's ever interested in partnering or has a creative idea, feel free to email us. We're open to many different things.
I will add the email address down below. Yeah.
Great, thank you.
Is this app available for free download?
Oh yeah, sorry. Community of Microbes is available for, I think, both Android and Apple products. And yep, it's free. And it's, if you go to the website, it will shoot you to the website where you can get the interactive PDFs, which allow you to use your phone to have them pop up. So it works in this digital landscape as well.
Oh, that's great. And I guess a big theme of your research is blending academic and industrial research. Could you talk a bit more about that? Because sometimes I guess researchers are associated with blue sky researching, whereas the research that you're talking about is all applied, all like used by the industry.
Yeah, so I think it's important to remember that some of my research starts off with exploring the microbial cosmos of paper wasp nests. It could almost be no farther removed from application than you could even imagine. And I like the puzzle of connecting the two. And so my mission in life is to reveal the beauty and utility of the microscopic world around us. And one way to do this is by not only proposing an application of a microbe, but actually showing to industry and business people and other audiences and stakeholders that indeed, if you find this microbe in a paper wasp nest, 10 months later, you can have a new commercial product, new beers that taste better, or better tools for brewers. And then I think in each one of these case studies, it then adds trust to the statement of, if we just discover another microbe species, we really can build a better future. And so broadly, I want to show that utility and beauty, and I will do it as a communicator, as an inventor, as an entrepreneur, or as an academic researcher, as long as we accomplish that mission.
With the microbes that we interact with, most of them are in our food and we can't see them. Is there something that we can see?
Yeah, so we've already talked about some of the microbes that exist around us that we know by their taste or maybe the aromas they're producing in bread. And there are some microbes that, though they are invisible as individual cells, we can see them with the naked eye. And one of my favorite examples is lichen. And so this is the plant-like material that you often find on trees or growing on rocks. And this is actually a biological castle. So it's a community of different species of microbes that are working together. And one microbe is providing the shelter, and another microbe is providing the food from photosynthesis. And this is such an effective partnership that these lichens have been sent to space and survived the radiation of space. That is a wonderful house created by a simple microbe. And so now when you go about your sort of walks to places that you go, you can notice these lichens and think that they may not look that impressive. They're just sticking on a rock, but they're able to endure unimaginable stresses, all because of the microbes.
Okay. Moving on to another question is, sorry, I lost the cue. Moving on to another question. So among fermented beverages, we know about beer, wine, cider, and another beverage that's making a lot of noise right now is kombucha. So is kombucha is very sour too, and is the sourness of kombucha coming from the same microorganisms in sourdough or yogurt, or what's the microbiology of kombucha like?
Yeah, so in a lot of fermented beverages or fermented foods, you'll note that tartness. And tartness, or that sourness, the sour of sourdough, the sour of kombucha, even the sour of yogurt, generally comes from two predominant acids. One is lactic acid, so that's where you're getting an acid in your mouth. If you plug your nose, you'll still get that tartness of yogurt. And another acid you're getting mostly in your nose, so that's vinegar or acetic acid. And in sourdough, you get a little bit of both, but typically more lactic acid. Yogurt, you're getting more of that tastes like acid, doesn't really smell like acid. And then in pickles or kombucha, you're getting more of those acetic acids, those volatile acids that smell like sourness. The thing that's really remarkable about kombucha, as anyone who has made it can attest, is that it creates this sort of plant jelly-like material at the interface of liquid tea and air, this mother, which is where all the microbes are living, and they're churning out what is basically cellulose or plant material that's actually microbe material. That material is so good at sucking up moisture that it's actually used in cosmetic face masks that highlight biocellulose as an additive. So that biocellulose is really microbial cellulose. And it's used, again, for cosmetic masks that help moisturize. It's also used in some wound care products that allow for increasing moisture. And so this little humble microbe that you can find in kombucha that's producing a lot of those vinegary notes provides a lot of other services to us as well.
I think that is a great example of showing the diversity again of these microbes, and that they not only can do one thing, but they are capable of doing many things at the same time.
Yeah, I think I love that part about microbes, that they're often sort of a Swiss Army knife of microbes that are involved in making wine are used by the coffee industry to help make different flavors in coffee, and microbes that are making kombucha are also making this material that's used in wound care. And before, I think I mentioned microbes that are producing xanthan gum, which is an additive in many foods and cosmetics. I mean, it's used, if you start looking through the ingredient list of foods in your cabinet, you'll find that it's used across the board in many things. So again, humble origins to our technology.
You mentioned the wildsado project that you are running with NC State. So we are doing a public science experiment here in Tassie, where we are following the same protocol and we are getting the data and we're going to contribute to the wildsado project. So could you give a few talk a bit about like the importance of the project or what we would get out of it?
Sure. So as a team, we are thrilled that you're helping out. We want to just mention that. We've been shooting back emails filled with enthusiasm and excitement about this.
We've got 50 participants.
That's fantastic. So this research came about, the origin story is a few years ago when we started a global participatory science project to understand what microbes are in sourdough starters. And so this is the sourdough microbiome. If anyone sampled their own gut microbiome to find out what species are there, we're using very similar approaches to understand what species are in sourdough. Is it pretty much the same two species, regardless of where the sourdough comes from? Do these species shift over time and generations as people pass down their sourdough starter? Do these species differ depending on if a sourdough starter is made somewhere hot and dry versus somewhere cold? We wanted to begin to uncover the answers to some of these questions. And so we collaborated with a number of universities. This is really a beautiful example of many scientists working together. And we haven't published that research yet, but it's coming up soon. And from that, we wanted to ask the next question, which is, what's the activity of these microbial communities and how do they shift based on where people are making them and what flour they're adding, other resources like that, when they're using the same protocol of starting that microbial sourdough starter. And so this project involves sort of a protocol of making a sourdough starter from scratch with water and flour and tracking its activity and then reporting those results. And then also using one of my favorite pieces of equipment in the lab, which is our nose, to understand some of the aromas that are coming from these microbes. And I say that somewhat tongue-in-cheek in that our nose is definitely going to be different among people, and it's not a calibrated piece of equipment, but indeed our ability to smell and detect different aromas is quite remarkable. We are better than the best GCMS or equipment that's available at detecting certain smells. And so while in the future we will get more granular and quantitative about these assessments, for now we're relying on the public to help us understand how do these microbial communities differ in activity as it relates to sourdough, and what are some of the other aspects that these microbial communities differ from, such as smell.
I think what you said with the nose is, makes it really for a nice public experiment. You're not only kind of fostering this, your own culture, but you always, it's another sense you're kind of using to experience this sort of experiment, which is, I think, not common in a lot of public experiments or, citizen science project that you actually use your nose. So I think that's quite neat.
Well, thank you. I think it's an important one to think about in terms of teaching observation skills to people as well, right? It's not just about the visuals, but so often we overlook smell. We don't have the same lexicon or words to describe smells, but they're so important to understanding our natural world.
Also, we are for the experiment here, we are using flour from Tasmania. Rye flour, organic wholemeal, and plain white flour, unbleached. But given the unique, environment, water, and flour in another place where you haven't done the experiment as of yet, are you expecting it to be very different, or is that something that can be expected?
So your question is asking the expectations of sort of different activity among different groups using different materials?
It's more like based on, say, a rye flour starter in America versus a rye flour starter in Tassie, where we are experiencing winter right now. So all of these aspects of air, water, temperature, flour, that's expected to affect the final outcome?
Yes, I think whenever we do participatory science such as this, we're relying on lots of people carrying out the experiment so that we can start to tease apart some of these factors that are also going to contribute to that activity. And from these results, we will then be able to do other lab experiments to follow up on some of the findings. So it won't be the whole story, but it will definitely help us get closer to understanding what's going on.
So, well, ending on a more general note, I'd say, is given like the current situation of a global pandemic, which has affected all our lives, what would you say is like the importance of science communication and awareness of science?
Yeah, I mean, science is a beautiful thing. It's helping us get closer to cures and vaccines to a deadly pandemic. Science is helping us understand what behaviors keep us safe. But I think that we've seen now more than ever, it also takes effective science communication, not just effective science, in order to change people's behaviors, to enable people to make the decision to use the vaccine, to wear masks, to do things that protect all of us. And so I think now more than ever, it becomes clear how we need to invest in science communication and understand that science communication is a set of skills that can be learned. There's evidence-based practices that we can adopt. And by being better science communicators, we are enabling people to use the science that we've worked so hard on finding.
Great. Thank you. Do you have any last messages to our audience? Last messages you kind of want to convey from this talk?
Yeah, so deep gratitude. You are all becoming microbial explorers yourself by learning about this microbial world. And it's a space where we are living in the greatest age of discovery of any humans that ever walked this earth. There are more species we don't know at the microscopic level than those that we do know. And you need to go no farther away than touching your tongue to find species that are unknown to science. We have not yet worked out ways of growing them in the lab to find out all that they can do. And so I urge you to continue your exploration, your readings, to find out more, and to share the beauty of this microbial world with others in your life. Thank you.
Very inspirational words. Yeah, I think before Deepon signs off, we will put all things, your work, links in the description below, so people can find you and your work you have done. And yeah, thank you very much for your talk. I think it was very interesting, inspiring.
Yeah.
Thank you.
Thank you so much